All right, we're going to get started. Thank you, everybody, for joining us for our first online meetup. And I'm going to tell you a little bit about our group. And this is the panel on transitioning from a wet lab into a dry lab. So getting to a more computational side of things when in your research. All right, I'm going to stop sharing my screen and I'll give you a few pieces of information as we keep going. Harry, again, I'll restart. Hi, everyone. My name is Radhika Kotani. I am the training director at the Harvard School of Public Health's Bioinformatics core, and I'm going to be the moderator today. So I wanted to give you a few pieces of information about our meetup group. So we started this meetup group, the Boston Area Women in Bioinformatics meetup group, was started by Lorena Pantano, one of the co-organizers now, in April 2015. And it held regular meetings supporting women in bioinformatics for the last, for about 4 years until last year. The group went on a hiatus for about a year. And it has been given a new lease of life with several new organizing committee members joining, also with help from Diamond Age Data Science to make sure that the meetup group was funded. So we wanted to highlight one thing before we get started and move too far into the panel discussion, that even though our name is quote unquote women in bioinformatics, we welcome everyone, all gender identities to join us in this meetup group and at our meetings, so on and so forth. We actually envision this group more as a support group that enables women, people from underrepresented groups, as well as allies to share their work, to share their ideas. And it's a bringing together kind of a comfortable space for sharing bioinformatics ideas and supporting each other. For this specific panel discussion meetup that we will be going through, I specifically want to thank my co-organizers, Katie Hughes, Eleanor Howe, and Sakina Saif, for bringing all of us together here and doing a marvelous job setting this up for us. And last but not least, I want to thank the 3 panelists for spending their lunch hour for some. We have someone joining us from California, so it's breakfast time, talking to us about their exciting career paths and how they made the transition and how they learned what they learned as they do what they do now. So what we will be doing moving forward is I want to talk to the panelists a little bit or tell you how things are going to go. And then I have a few tips for the audience. So for the panelists, we will be giving each panelist 3 minutes to introduce themselves before we jump into questions. And they've been informed that this is what's happening. So they'll give us a 3-minute discussion, 3-minute introduction. And panelists, do keep in mind, I am using my phone to keep time and might interrupt you close to the end of the 3 minutes to make sure that we keep moving and to enable a full and diverse conversation. So apologies in advance if I interrupt you. For the audience, thank you again for joining us. We will be encouraging you to bring up your questions for the panelists and questions that you want discussed. But we want to make sure that we do it in this as efficiently as possible. So if you can post your questions in a direct message to either Eleanor has a questions next to her name, or Sakina, who has a questions next to her name also, and they will be passing on the questions to me. And as and when it seems pertinent, I will be slipping those questions into the discussion. Lastly, we ask that you mute yourself and keep your videos off during this panel discussion. That way, as we are walking through the panel discussion, the 3 panelists and the moderator are the people who are showing up as we speak. rather than it being a little more chaotic. So we appreciate everything that everything that you guys all bring to the table. So thank you again for joining. All right. So I'm going to get started with giving the panelists a few minutes to talk about themselves. So if you just give me a second, let's see. All right. I'm just getting all my questions in a row, or my ducks in a row, I should say. All right. So we are going to get started with Joohyun, who's going to tell us a little bit about kind of her background and how she learned about bioinformatics and how she made the transition. After Joohyun, we'll go to Tan, and then after that, we'll go to Mary. So Joohyun, if you want to get started and tell us a little bit, 3 minutes, tell us about your career trajectory and what you do. Thanks.
Sounds great. Thank you for the introduction, Radhika. So yeah, as she said, my name is Joohyun. I'm a senior scientist in computational biology at Kintai Therapeutics. It's a Cambridge-based biotech company, which it's a startup, but no longer a small startup, I guess. So I will go in a non-chronological way. At Kintai, we study connections between human gut microbiome, gut immune system, as well as enteric signaling network. And we essentially look for small molecules that could impact diseases in oncology and urology. I, as a computational biologist, particularly study how human gut microbiome shapes the progression of various types of cancer, as well as how patients respond differently to cancer therapy. So I do this by utilizing or analyzing diverse sequencing data that either came from in-house platform, as well as that we downloaded from public domain. As for my little bit of background, so I have a bachelor's in biology and PhD in genetics. So my training in bioinformatics mostly came from taking courses and conducting my own thesis work during graduate school. I was interested in making transition probably at the end of my college years, and subsequently when I was working as a technician in the lab. Rumor has it that I was making RNA-seq libraries at the time and heard that there is a robot out there making RNA-seq libraries. So I was afraid that I'm going to be replaced, and hence I started programming.
Great. Thank you so much. Thank you so much for that really clear introduction. Tang, if you want to go next. I'm sorry if I'm not getting your name perfectly right.
It's not. It's correct.
Can you, if you can say it, that would be great too.
Okay. Hi everyone, my name is Tang, and I am currently a bioinformatics research associate at Bioinformatics located in north of the Bay Area. So I got my bachelor's in biochemistry and molecular biology, and then straight out of college, I worked at an antibody company called Cell Signaling Technology, and there I was 100% wet lab. And fortunately, I was lucky enough that at the company, I found out that we have a group called the Bioinformatics and Data Science Group, and which piqued my interest. And so then the group leader, and he's also the bioinformatics director for the company, he allowed me to be unofficially part of the group. So I got to drop in on their meetings, and I also got to do little side projects with it as well. So that gave me insight into the bioinformatics world. And while at Cell Signaling Technology, I pursued my master's degree in bioinformatics. And I also did my co-op at Cell Signaling Technology as well, and that really helped with the classes that I was taking, and I got to have a hands-on experience and applying what I learned into the real world. So that really helped as well. And after getting my master's, I moved over to California, and I'm now doing computation 100% of the time.
Great, thank you so much. That's really great, and both of you have stayed totally under time. So next we're going to go over to Mary Piper.
Great, thanks, Radhika. My name is Mary Piper. Currently, I am a research associate and associate training director at the Harvard School of Public Health Bioinformatics Core with Radhika. My route into doing bioinformatics is a bit circuitous. I started with my undergraduate degree in biochemistry and molecular biology. I went on to get my PhD in virology, so I did virus research for my PhD, all wet lab. After my PhD, I pursued a master's of public health in epidemiology with the idea that I would be an outbreak investigator. At that point in time, my husband was finishing up his PhD, and we were moving, and he got a postdoc in Boston, and I naively thought that I could easily get a job in public health in Boston, which didn't end up being the case, but it was fortuitous all the same, and that I did get a job at the Broad Institute, teaching for one of their summer programs, so teaching genomics as a summer program. And that kind of opened my eyes to the excitement of next generation sequencing, and kind of fueled my passion. I had a background in teaching from graduate school, so I taught throughout my graduate school. And it was also something that I enjoyed. And so when I heard about the position at the Bioinformatics Core at Harvard being available, that was mainly looking for teaching experience. I didn't have any background in bioinformatics at the time, but I applied, and it turned out that they were willing to train me on the job. So for the last 5 years, I've been at the Bioinformatics Core, doing training and consulting as well.
All right. Thank you so much, Mary, and thank you to all three of the participants, panelists, to staying within the 3 minutes. Actually, all of you stayed within the 2 minutes. So this is great, and we have some questions lined up for you guys, and we'll sort of do a round robin. And if you don't have much to add to that question, just say I'll pass on the question. So I'll start kind of with one of the basic things, and this is more a question for Joohyun and Mary. Would you recommend that a PhD is necessary for someone who wants to do bioinformatics? And kind of, if you can, that's a broad question, but kind of more focused would be, are there things you learned in your PhD that are helpful to your current, whatever you do at this moment in time? So maybe Joohyun, you can go first.
Sure. Perhaps since I did a PhD, perhaps I am a bit biased. I really enjoyed the fact that my full-time job, essentially in grad school, was to learn new things. Mm-hmm. And that really helped me gain foundation to work with lots of things in condensed time. And certainly having a lot of research experience helped me get good at both biology and computational skills. And that certainly helped me when I got out to look for a position where I could direct and shape research. That being said, there are some cons associated with getting a PhD, correct? So there are lots of, your life will be affected in one way or another. In my case, I took a pay cut to go to grad school because I was working prior to attending a PhD program. I had to also move, which I think a lot of people have to move for a degree program. And it's also a lot of stress that comes with the program. So it's certainly not for everyone. I think if you're interested in driving and shaping research direction and project direction, then perhaps doing a PhD is something that you should consider. Mm-hmm. But if you're fine with following what is laid out, I think you will be just as successful without a PhD.
Okay, great. Thank you, Joohyun. Mary, do you have something to add to that?
Yeah, no, I agree with what Joohyun was saying, but I would add that in my experience, the PhD has been really helpful in a few different ways. So while I think you can have a successful career with a master's as well, for the PhD, for me, I think it helped me make the transition. Like coming from the wet lab and having diving deep into research and having that background in science, develop the confidence that you can tackle any new project and know how to, like, you have the toolkit available to address it. So I think that's one thing that the PhD helped me with, is that I had that toolkit that was accessible, and it gave me confidence when trying to transition to a new field. The other thing that the PhD is definitely given me is a greater opportunities. So both my position at the Broad that I initially got, as well as my position at the core, required a PhD. And I know that you know, my options would have been more limited had I not had the PhD. And so in that respect too, like, there are opportunities, there are good opportunities for people with masters, but a PhD will give you a greater breadth of opportunity. And that goes like across disciplines as well. Like, even though my PhD might not be in bioinformatics, right, it's the fact that I have a PhD, that I've done that depth of research, that I have that toolkit, is something that a lot of employers look for as well.
Yeah, great. Thank you. So, Tang, you don't have a PhD, but you got a bachelor's, and you've gotten a lot of hands-on experience. And based on what you said, you had some supportive leadership in the company you were at to help you get started. And how has this worked out for you? And how important do you think the co-op was to help you make this transition?
I thought the co-op was really, really helpful because for bioinformatics, I realized I learned the most by actually just diving in and doing it, learning from my mistakes, and using the programming languages I was taught and applying that to a real life problem. And by doing that, I learned a lot. And I guess it's just that once you get right into it, you just and get the hands-on experience, and it's the application, being able to apply what you learn into a specific problem. And I thought that really helped me a lot and gaining experience.
Great. And so we have a direct question related to that. Do you think that this transition would have been possible without your master's studies in bioinformatics? So after your bachelor's?
I think that without pursuing a master's, I think that I probably can still be able to gain the bioinformatics experience, but it'll probably be at a slower pace. What really helped was working at the company. There was a bioinformatics and data science team, and with that, they were very supportive, and they would let me do side projects if I wanted to. So that really helped a lot. So I would say that no, you wouldn't need a master's program in order to kickstart bioinformatics, but it would probably be at a slower pace than if I weren't to pursue it.
Yeah, and it sounds like a lot of your experience at your company also was, in a way, similar to some aspects of a PhD, where you were allowed to pursue your own interests, your research interests, which I think also helps in building that confidence that Mary was talking about.
Yes.
Transitioning. Yeah, that's great. So perfect. Thank you all three for answering that. So now my next question is related also, of course, to the transition. How difficult was it to transition to a fully computational role? Did you face any kind of obvious obstacles? And did you miss the wet lab? That's one question I get a lot. So I'll start with you, Tang, since you just answered.
I was, when starting out in the master's program, a lot of people were asking me, is like, oh, like, are you going to stick with wet lab, or are you going to transition completely to the bioinformatics? And having experience in both, I realized that I love the computational aspect of it more.
Mm-hmm.
And in the beginning, I thought I could split in half and do 50-50, but I come to realize that I wanted to completely devote my time to computational biology, because I realize it's really hard to switch your brain back between wet lab and computational. It's like, especially when you have to go back and forth if you're running experiment, and then you gotta let it wait, and then you're like, okay, I'll go back and do some programming, and then you gotta get away from programming, and it's like you have to shift your mind.
Context switching, yeah.
Yes. And I realized it's like so complicated going back and forth. And so I was like, I think it'd be better if I just focus on one. And I realized that it was the computational that I really enjoyed the most.
Okay. And did you face any like major obstacles when you made these decisions or transitioned that you felt like were difficult to overcome?
So since I started the bioinformatics program without any programming experience, the beginning was rough, especially when I started my project during the co-op as well. It was a big learning curve, because you had to learn a lot of new things, especially it's like learning a new language.
Right.
So it was rough in the beginning, but then it's once you just keep going and keep learning and ask a lot of questions, especially with your mentors. They're there for you. They're there to help you. So always keep an open mind. And even though sometimes it does look like you're spending like 2 hours on just solving like one simple thing, you couldn't figure out what it is, it's okay. It can get frustrating, but just go, it's like, just realize that you will solve it, and then there will be people out there to help you as well.
And it can take more than 2 days sometimes, or 2 hours. It can take more like 2 days or 2 months.
Yes, it can get frustrating, but at the end, it's really worth it once you do solve it. I guarantee it you.
Yeah. Thank you. I'm going to pass it on to Mary. So just to reiterate the question, how was the transition for you? Did you face any major obstacles? How did you overcome any of these? And, anything else you might add?
I missed the wet lab.
Yeah, you missed that was it. Thank you. Did you miss the wet lab?
The wet lab, I thought I would, but I had done research for 3 years of my undergrad and for 4.5 in grad school. And by the end, I was tired of cell culture. And, I thought that I would miss it more than I did. So that hasn't really been much of a problem for me. I love the science, so the science is what really gets me excited, and that's still there in the computational analysis, and I feel like that's kind of one of the key drivers for me, because you get someone's already done all the prep work, and you get to hand them the answers. Like, these are the genes that might be really important to your disease. These are the processes that might be contributing to this particular condition, right? And so the excitement for me was the science, and I feel like that's almost heightened a bit when I do computational work. So I haven't missed the wet lab, but I was tired of it when I left, so there was that too. The other question was the transition. How difficult was the transition? It was a very steep learning curve, so I'd say similarly, I was learning on the job too. So like I hadn't any background in programming. I was a bit intimidated because I wouldn't have considered myself a computational person, in that I didn't know everything there was to know about computers. And I think my misconception going in was that might be something that was necessary. And that's not the case, but that was a misconception that I had. And so that was a bit intimidating for me. The other thing was I had to learn, R and the command line and all these workflows for next generation sequencing analysis. And that's a very steep learning curve. And so during that time that it was taking me to learn those things, I'm at my job where everyone else knows these things. So that was a bit isolating, but also I felt like I wasn't contributing enough at my job because I'm learning, right? And it was understanding, my job was understanding of that, but it still was a bit isolating and difficult. And it probably took me like a good, 6 to 8 months before I was at the point where I could actually contribute substantially to both the training and to start consulting.
Yeah. Thanks, Mary. All right, Joohyun. So obstacles when you made the transition, or did you miss the wet lab? Do you want to highlight anything?
Sure, yeah. So I still do miss the wet lab. I think so throughout my grad school career, I started off in the wet lab and then slowly transitioned to the point where at the end of maybe last 2 years or so, I was basically out of the lab. But lab is such a comforting place for me that I would occasionally go in and do little things just to keep my interest in biology rooted, as PhD can get frustrating at times, or with any projects. In terms of transitioning and having difficulties, every step of the way, I feel like there were challenges. I think one thing that I would mention, I completely agree with what Mary and Tang had shared with us. And I think another layer that I would add to that is the fighting with or convincing myself that I am capable of doing this. I never thought about imposter syndrome until I got to grad school, and it's something that I have been dealing with since. And when there are not as many people in, let's say, bioinformatics class that look like me, or in the workforce there are people who look like me or who are women, it can get intimidating, even though there is no one who is directly telling me that I'm not able to do this job. So I think convincing myself to say like, hey, I can learn these things, I am a quantitative enough person to achieve this, has been one of the struggles that I had to deal with.
Yeah. Thank you so much. This is very interesting and insightful, and sounds like you all had some similar challenges, but some challenges were definitely unique to the situation you have found yourself in. And I think it's going to be unique for people wanting to make this transition based on their backgrounds, their personalities, the work environment they go into as well. So this is very valuable. Some of the questions we are getting related to some of these things, and we had it on our list as well, were kind of, people are asking what should they learn as far as programming languages go. And I think Mary and Tang have already talked a little bit about how they felt like that was one of the obstacles they faced, steep learning curve with the programming. So, if you wanted to recommend to somebody that before you start kind of full-fledged working on something, are there any specific languages or tools that you would recommend for them to start with? So I'll start with you, Joohyun, for that question.
Yeah, my answer may not be the most conventional answer, but
hey, that's why we have this diverse group of people here.
Yeah, so personally, I use R as my main weapon of choice. I am able to read Python and write a little bit, but I certainly feel more comfortable using R because that was the language I used throughout the past few years as I was doing my work. The unconventional part is that I think I did not see the importance of statistics until maybe halfway through or through my transition. The knowledge in programming languages and packages to use will change because as research goes on, there are lots of things that will be updated, and then whatever that I used to use for RNA-seq analysis are no longer the first tools to go, for instance. But math and statistics change little, relatively speaking, and that will really help you go a long way. So I would say, sure, learning how to program is important, but having a foundation in statistics and basic understanding of math will certainly help you move forward.
Yeah, that's a great answer. Yeah, thank you. I think that's probably a little more conventional than you think it is. So I'll pass it on to Mary.
So similarly, I'm coming from a working environment using next generation sequencing. So I am aware that there are other types of bioinformatic analyses that other people might be doing. But for next generation sequencing analysis, R is definitely is the language that we need for a lot of the tools that are necessary for RNA-seq. So our best practice tools are in R for doing a lot of the analyses. So R was crucial to learn, as well as the command line. So, and along with the command line comes some type of cluster or environment where you can access, large number of resources. Mm-hmm. So learning the command line, being able to access, high amounts of memory, high amounts of space to store your data, things like that is really essential. And along with that comes data management practices. So I know you just asked about programming languages, but those are also kind of combined with it, along with the statistics. And I'd agree too that statistics, I kind of wish I remembered more of the statistics from my public health background. I have forgotten a lot of them, and I think that they help you understand what the different tools are doing a lot more. Mm-hmm. But if there's two that I would suggest, I would say R and the command line. There's a lot of people in our core, me included, that don't have a strong grasp of Python. Mm-hmm. The people in our core, or the people in our group that work with Python, tend to use it for pipeline development. So mm-hmm. if you're looking to do development, Python would be important. If you're looking just to do analyses, it kind of has a lesser role. There are tools that can be helpful in Python to some of your analyses, but the main ones that we use are in R and the command line.
Great, thanks, Mary. Tang.
Okay. So I agree in that statistics is very fundamental to bioinformatics. And even till now, I'm still keep refreshing myself on statistics just so I can stay in being able to analyze things. And I'm going to say I'm pretty biased because I actually started off analyzing my co-op project using R, mm-hmm. and I've grown very comfortable with R, and I've grown to love R. So R would be my number one go-to language. But I also want to add that I think depending on the language, it's based on what questions you're trying to answer. Mm-hmm. Just like what Mary has said, maybe like Python for pipeline development and then R for analyses. So it really depends on what kind of specific questions you're trying to answer and going from there. But I want to say that R is probably because I'm biased to it.
Yeah, that sounds great. Thank you all. And I think I've spoken to a lot of people who do this, and I hear pretty much very similar answer. If you talk to somebody who started out using one language, and if that language, it's possible to keep going with that language, they will. R has been one of the big ones, at least for doing next generation sequencing based bioinformatics. But Python is great for development, and people use it for all sorts of things. So along those lines, I have a question for you guys, which is, how do you stay, this is an audience question, how do you stay on top of your skills with the number of new tools, the number of papers? What do you do to make sure you have the time not just to read the papers, but to learn new skills? So I will go with you first.
I guess by staying on top of it, because since I'm working in the bioinformatics field, so based on the projects that I'm doing, I get to apply different things. And if there is something new, then I try to apply it. And then also I have people in the workplace as well to help me and to give me guidance if I do need help.
Cool.
So just by learning new things, it's just trying to apply that into your real life applications.
Yeah, that's the best way to
Yeah.
retain what you're
Mm-hmm.
learning, right?
Mm-hmm.
Yeah, makes sense. Great. I'll go with Joohyun, and then I'll come back to Mary on that question.
Yeah, so similar to what Tang had suggested or shared with us, I think working with others is certainly the way to go for me. I rely on collective intelligence to tell me what's new, and then I try to do the same otherwise. This has been especially crucial in working in a company setting where there are lots of meetings and deadlines to meet that are kind of tighter than what I was used to when I was in academia. So relying on other people to kind of tip me, and then vice versa, certainly has helped me understand what's coming up, what's new, what I need to pick up new.
Yeah, great. Mary.
I will reiterate all of that. I think in that I work at the Bioinformatics core, and so as a group, we try and stay on top of it, stay on top of the science. And there are different social media areas that we use, like Twitter. We follow important, large, the big names in the field, kind of the people making the tools, the people updating the methods. They often have Twitter. I'm trying to get better at using Twitter. I am not very good at it. But a lot of my colleagues are, and they keep me abreast of developments and things like that. And of course, following the papers, oftentimes we have a Slack channel, right, where we're posting papers that are new. And whenever we're doing a new technique, we'll kind of like kind of like what you do as a research project, right? You'd investigate what the current applications are or the current methods for that tool. There might be some benchmarking papers that have benchmarked the tools against each other. Again, your friendly colleagues that can help you, which tools they recommend, why they recommend them. And then sometimes you do your own benchmarking. Sometimes you try a couple of different tools, and then you share that information with your colleagues. And sometimes they do the same and they can share it with you. And I think networking within the community is really important as well. So yeah, it is hard though. It is hard to stay abreast of this current, like, rapidly changing field.
Yeah. Thanks all three of you for answering that. I have a related question that I think I'm going to start with Mary on. What do you think is the most important? I say most important. You can give me more than one. Soft skill. So not a technical skill, but a soft skill that you need to be a bioinformatician. But also, if you want to talk about technical skills, that's good too.
So communication and organization. Communication would probably be the first, in that a lot of the scientists that you're going to be working with do not have a background in the methods that you're using, and to be able to relay to them the importance and kind of the conclusions, have them be able to interpret the results, I think is one of the most important things of our job. Because you need to make sure that they're interpreting the results properly. Also, you have to be able to communicate that verbally as well. So not just in your reports and things that you're giving or that you're providing the scientists, but also be able to communicate that either through presentation or just one-on-one is very important. So the people doing the bench work can understand the results that you're giving them from the computational work, and then can incorporate that to progress the science.
Yeah.
So I'd say communication would be the first one, and then organization. There's so many files and so much, like for the directory structures that you're creating, the files that you're generating, the results that you're generating, all the output. There's so much organization that's required that if you don't have good organizational skills, you'd never be able to reproduce your research. So I feel like those first, communication and then organization, are my top two.
Cool. Thank you, Mary. Julien, do you have things to add to this? soft skills that you think have been useful for you?
Yeah, I would also put communication on top. That has been very crucial. Specifically, I've seen scientists who create really easy to read, easy to understand visualizations moving forward more so than other scientists who are less able to do so. So I think communication, understanding how to deliver your results to other colleagues who are not in your field, right, and in a more straightforward, intuitive way is very important. Another thing I will add, maybe collaboration or ability to collaborate well with others. I think in that regard, we're actually really well suited because we all have experience working in the lab. So when you talk to the colleagues who are mostly based in the lab, I think your experience will serve you really well and help you collaborate better.
Yep.
But especially in the industry environment, I'm sure, as always with everywhere else, but I think there has been a heightened emphasis in collaboration in this industry experience I've had.
Right. That makes sense. I think we are, in a way, from going from wet lab to dry lab, we've suddenly become interdisciplinary, which makes it makes communication, networking, and collaboration all really important and present. All right. So Tang.
Okay, so sorry, I'm going to be a little bit redundant here, but I completely agree with communication and just not being able to explain your results, but also with communication, you can also learn from your colleagues as well. So it goes hand in hand. And so I completely agree with communication, and yeah.
Great. I think one of the underlying things that's coming out of here with all of these things is also teamwork. I've heard all of you say, you've learned a lot from people you work with and all of these things. So teamwork, which, all of these are overlapping skills. So yeah, thanks. So next I had a question about, so we have an audience question. And this is, I think, more for Joohyun. Someone was asking how was it doing like a wet lab and dry lab 50-50 split sort of during your PhD? And is it like, if you have a dual wet lab, dry lab status, do you have any wisdom about making sure that you get it done and don't get bogged down, or any thoughts you have, Joohyun, on this?
Yeah, so I guess I'll clarify a little bit more about how I made the transition. So I don't think there was a point where I was really split between 50% of my time in the lab and then 50% in bioinformatics. So my PhD project was actually using RNA-seq, understand how innate immunity works. So I would work with fruit flies and I would infect them in the lab, and I would collect their samples over time and then made RNA-seq libraries. So that was mostly on the wet lab side. So once that part was done, I mostly stayed in the dry side, in the office side, analyzing the data myself. So I didn't really manage 50-50% time myself, but I'm trying to see if I could remember some of my colleagues who had to do that. And I think some found it really refreshing because they, let's say, got stuck with whatever programming problem that they're trying to solve, and then they would just redirect their frustration into the lab by getting something like running a gel or something like that, and then vice versa. So some found it refreshing, others found it difficult because their attentions were divided. Yeah, that's, I think, to the extent that I could share.
Yeah, great. No, that's very helpful. Thank you so much. And we have a few more audience questions. There's a question about asking for resources, and we can post some resources on our meetup group, comments for this meetup afterwards, and we can ask the panelists to send us their recommendations for learning resources as well, that we'll make sure to post. So that was one of the questions. So there was also a question about getting pigeonholed. Do you find that as your career progresses, you are getting more pigeonholed into certain types of projects, that you would like to be more, have more diverse set of things that you want to do, but you're feeling more pigeonholed? Or the other way around, or kind of, if you were feeling pigeonholed, how did you kind of find yourself? How did you transition out of that into a more productive way of working? So I'll start with you, Joohyun. Or if that's not relevant, that's okay too.
Yeah, that's an interesting question, because I felt that way a little bit in the middle of last year at work. And one thing that really helped me was that I communicated that to my manager, saying, hey, I am doing XYZ work, but I feel like I'm doing the same thing over and over again. I'm interested in learning new skills. Would you be okay if I take some part of my day off and use that time on, let's say, taking a new course in Coursera? Mm-hmm. So, and he was fully on board with that. So for, I think, 2 months or so, I spent Friday morning working offsite, just churning through course, and that really reignited my passion for doing the work that I do. So that was certainly how that really helped me in getting out of the pigeonhole.
Yeah, that's great. Tang, do you want to, do you have any thoughts on this? Have you felt like this?
Yes, when I was after I graduated from my bachelor's and started my full-time position, to get out of it, I actually asked around and looked at the other groups in the company and see if I can maybe shout at them, or I can help them out, or I can learn a different skill from them. So, because in the beginning, yes, I did feel like my work did feel redundant, but then just by learning new skill sets from other groups as well, that really helped me to get a different experience.
So you took the initiative to ask people about learning new things and stuff like that. That sounds great. Thanks. Mary?
I haven't felt too pigeonholed, partly because of the nature of my job. One is that I do training and consulting, so that is constantly going back and forth. But then the other thing is that as a consulting core, we have lots of different projects, so it's almost the opposite in that sometimes you'd like to focus more of your energy on a particular project, but you don't really have the time to do that. That being said, I have talked to the director of our core too, and asked to learn new things, and they've responded well to that. So I think if you are starting to get bored, hopefully you have an understanding boss, and that there's someone that you can talk to about finding new and more exciting things that you might be able to add to your repertoire.
Yeah. Sounds great. Thanks all of you. So we have about 10 minutes left. And if there are additional audience questions, I invite you guys to post them, and Eleanor and Sakina will pass them on to me. And while you're doing that, there was a question that I'm going to kind of reframe a little bit. So all of you transitioned from the wet lab to dry lab. You have you spent a lot of time in the wet lab doing, either virology or doing things that were more related to sequencing or upstream of the data analysis that comes with bioinformatics. What do you think about someone pursuing kind of pursuing a degree in analytics and data science, kind of a much broader informatics space or data science, quote unquote, space, rather than specific to bioinformatics or research in bioinformatics? Do you have any thoughts on that in your experience? What do you think of, I don't know, Mary, I'll start with you if you have any thoughts on this.
My thought would be, getting a degree in data science would probably make you more like available for a larger diversity of jobs in other areas, particularly in bioinformatics. I know at our core, if you didn't have any experience doing like or knowledge of like next generation sequencing analyses, that it might be hard to get a job with us specifically. So if you're doing data science, but and you don't have a background in doing the types of analyses that we do at our core, then you probably wouldn't be able to get a job in that specific area. That being said, data science degrees are good for a lot of other areas. So I guess it depends what your goal is, your end goal. Sorry, I don't know that was very helpful.
But. No, but that is, that's also good to know. As being someone in there, like that's kind of your experience. So, sorry, I'll go with Tang. If you want to, if you have any thoughts on this.
No, I agree with Mary in that if you were to pursue data analytics, it would give you a broader range in terms of like job prospects, and it really depends on what you're looking for as a career-wise.
Yeah. All right, and Joohyun?
I would echo what they have shared.
Okay, great. That makes perfect sense. Thank you all. And I had one more question, which, let me see. Oh, I had a question for all three of you, which was, this is my own question. You guys transitioned from wet lab to dry lab. How do you feel about instead of having transitioned, instead of having learned biology and then gone to do more computational work, if you had started with computational work and in bioinformatics, do you think the wet lab portion, and I know some of you have already alluded to this before, the wet lab portion, how valuable was that in what you do currently? And Joohyun, I'll start with you.
Yeah, so I could give you a concrete example. So some of my work support our oncology programs, and that means that there are scientists in the same team who are mostly based in wet lab. I am, because of the nature of my work, my analyses are mostly done in the early phase part of the program, where we do the analysis and we find candidates. And then based on that information, then assays need to be developed so that we can verify in the lab if our candidates are indeed effective. Because I have experience in some of the assays, if I don't have, or if I haven't run it myself, at least I have conceptually have some ideas about how they work or what they're targeting, it has been easier for me to talk to people who are developing assays, mostly in the lab, and also help them understand how to interpret the data better. So I think in that regard, having that experience has certainly made my job easier and also more effective.
So would you say it's kind of like you speak their language?
Right. And I think they also feel, well, I don't know, I haven't discussed this with my coworkers directly, but I think they also feel kind of affinity towards me because they know that I was also in the lab. They know that I have done Western blot or running a gel or PCR. So I think that certainly helps us bond better too.
Okay, great, thanks. Mary?
I would definitely second what Joohyun said, in that you speak the same language, you understand their experiment better. And we deal with a lot of different biological questions at the core, and I feel like sometimes I'd be lost or I wouldn't understand the significance if I didn't have that background that I do. And similarly to what Julien was saying is that, I can also make sure that they validate the results properly. Like we can talk about validation, we can talk about the type of tests that they are doing downstream to make sure that they're not just relying on the computational results that I'm giving them. They can tell me their experimental question. I can make sure that the analysis is tailored to the direct question that they want to address. so I feel like it's been such a vital component of doing my computational analysis, having that scientific knowledge. And again, as I said before, that's what excites me about doing the computational research, is kind of the biological results of it. And so I wouldn't have that gratification either if I didn't understand the underlying biology.
Yeah, that's great.
I agree with them both. It's
Okay.
having the experience, and then that makes it more understandable in the addressing the problems. And right.
Yep. Great. So we have a couple of minutes left, and there were a few questions. There are a few more questions coming in. There was a question about, where do you find good mentoring to learn computational work? And it seems like our panelists all took different paths. One of them learned on the job. Two of them learned on the job in a way. One of them learned during the PhD from various different mentors. And maybe, if we get a chance, we'll keep track of some of these questions and see if we can request the panelists to see if they'll answer some of them, and we can post them on the comments board, just so we don't want to run over time. So just wanted to say thank you, everyone, for attending today, and especially many thanks to the panelists for making the time and sharing their experience, their expertise. This has been wonderful. Thank you again to some of our co-organizers for putting this together. Just to highlight, we have another event coming up in July, and we will be announcing the date soon. This will likely be another online event, but we are also talking to other meetup groups about collaborating and coming up with something that's exciting, something that's useful and valuable. And details are coming by the end of this month, so watch our page. And thanks again, everybody, for joining us. This was a really great conversation. And have a wonderful day and a wonderful weekend. Thanks, everybody.
Thank you so much.
Thank you.